Hello, I'm Ushma Neal with the Journal of Clinical Investigation, and I thank you for joining us for another in our series, Conversations with Giants in Medicine. Today we shift our format and I bring you not one, not two, but three giants in medicine. Today I'm lucky enough to be joined by Dr. Jesse Roth of the Feinstein Institutes for Medical Research, Dr. Ron Kahn from the Joslin Diabetes Center and Harvard Medical School, and Dr. Jeff Flier from Harvard University, who served as the Dean of the Faculty of Medicine. We bring them together today as part of the JCI salute to the 100th anniversary of the discovery of insulin. Roth, Kahn, and Flier all played critical roles in the discovery and description of the insulin receptor and in outlining its critical role in diabetes. We welcome them together today to hear more about the heady days back in the 1970s while they were making their groundbreaking research discoveries. Welcome, Dr. Roth, Dr. Flier, Dr. Kahn. Thrilled that you were able to join me this afternoon.
Our pleasure.
Happy to be here.
Well, Jesse, I would love to start with you. Would you mind setting the stage for us? You are the head of the NIDDK at the NIH. You are running a lab together with Ira Paston, starting to work on cellular membrane receptors, but they aren't necessarily accepted outright in the scientific community. So before Ron and Jeff joined your lab, what was the field looking like? What were the issues that you were facing?
The story here really starts on July 1st, 1963, when I showed up at the NIH. And they were interesting times because if you were an MD, it was at the time of the height of the war in Southeast Asia, and people were frantically trying to find U.S. government jobs so that they would not get drafted and sent to Vietnam. It was at a time when the popularity of the Vietnam War was plummeting, and so I was lucky. I got a job at the NIH in an endocrine group, a very powerful and very good endocrine group. And when I showed up, the new boss, and it was funny because I had worked the previous 2 years in New York with Yalow and Burson. Yalow, you all know, won a Nobel Prize for her work on developing a measuring technique for insulin. And had Burson lived, he would probably share that Nobel Prize with her. But anyway, when I was at the in the Bronx, VA, where Burson and Yalow were, I worked on growth hormone with a colleague, Shimon Glick, and we were lucky. The work on the growth hormone that we did on developing growth hormone assays exploded, and that really copped for me the job at NIH. And I'm going to start with a little bit of a life lesson for the young people, because what happened when I came to the NIH, my boss, Ed Rawls, said to me, "Well, Jesse, what do you do when you get here?" So the logic of those days was if you had a little hot number, you then did it for the rest of your life. And that was the standard. And he said to me, yeah, you could do that. You could just do growth hormone for the rest of your life. But, these are special times. This is a special place. Think about something exciting you might want to do besides that. And so I met Ira Paston, and the two of us worked, and we decided, why don't we see if we can work on a method to measure insulin and see if we can see what insulin does, because really once insulin got to the cell, people didn't quite know what was going on. And in fact, a good way for me to tell you what the status of insulin was in those days was to take out an autobiography of Roy Vagelos. Many of you heard of Roy Vagelos. He's the president of Regeneron. He had been the chief at Mark Chevron at Merck. And then before that, he was a chair professor, a very important position at Washington University in St. Louis. And he, in his autobiography, he recreates what the theory was about how insulin worked in those days. And he says it beautifully. Everybody thought that insulin immediately went into the cells, found enzymes that it could bind to, and became coenzymes for their action. And we know that that's absolutely incorrect and not the way it worked out. But that's where Ira Paston and I started out. How does the cell know that insulin is there? And so what we did was to start to label insulin with radioactive iodine. If you did it like Bersaniello did for radioimmunoassays, you really beat the heck out of that insulin, and it didn't do well. It would bind to too many things. It would hardly bind to receptors, and that didn't work. So we decided we'd have to get a new method for labeling, which we did. And the other thing is that tissues, once you start to break them up, they don't like it either. So what we had to do was to develop a way to get insulin that was very gently labeled. And we had to do the same thing with the tissues. We then spent time developing methods for labeling insulin and have it retain biological activity. And the way we did that was to label only a small fraction of the insulin, but then separate the iodoinsulin from the non-iodoinsulin. And that took a little bit of time, but it worked. And the same thing with the tissues. If you broke up tissues, they didn't like it. If you worked with whole cells, that worked well. And in fact, we had to be a little bit odd about it. If you started out with the popular insulin-sensitive tissues like fat or muscle or liver, it didn't work so well. But if you went to blood cells, ooh, that was good. Nobody expected insulin receptors to be on the blood cells, but they were. And they were perfectly good receptors, and they were good heralds of what the other receptors were doing. And so we managed to get a method that we devised for creating a bioactive insulin that would label well, and for getting cells that would find these insulins just perfectly well. Now, we had other surprises. We thought that could we find situations where receptors for insulin were up or down? And sure enough, we did. So in patients, we started to study receptors on cells from patients. And in fact, they seem to reflect well the pathology of the disease.
Well, Jesse, I'm going to stop you there just because I think we're going to come around to the patients later on.
Okay, so I'll tell you what, Ushma, give me a short little one thing, which is a life lesson for the young people. What happens if you are lucky, like we were, and we stumbled upon a method that was really, really so promising? How much of your time should you spend on this vision you have of a method that's surely going to work? And most young people say, oh, of course, 100% of your time. Well, we're going to advise you to only spend about a third to half of your time. And the reasons are that, first of all, you may not be right. Secondly, even if you're right, it may take, and it often takes, much, much longer to work and get it to work. And then finally, when you get it to work, the acceptance of the ideas by the public can be very slow. So leave yourself a time cushion when you plan how you're going to get this fabulous piece of work to really come through.
So that's a perfect segue into asking Ron about when he arrived in Jesse's lab at the NIH. You spent about a year working on something that didn't work so well.
Right. Yeah. So this, as Jesse pointed out, was a remarkable time at the NIH in general, and in his lab in particular. My predecessor in the lab was Bob Lefkowitz, whose name is well known, I'm sure, to many people who are going to watch this, as a Nobel laureate who did really important work around beta-adrenergic receptors. But at the time, before I got to the NIH, he had been in Jesse's lab working on ACTH receptors. And Jesse was, as always, very excited about this new work that was so promising. And when I came along with another fellow, Art Sober, he said there was plenty enough for both of us to do. So we did actually take on this project. But I think that one of the things that Jesse already kind of alluded to is there was, in lab work, there is a lot of, I'll call it a little bit of luck of the recipe. You have to, this was a day when you did everything manually. And I think that there were no kits for anything. There was nobody, you couldn't go buy the labeling kit. You couldn't go buy the cells. You couldn't go do anything. Everything was done individually. And of course, that was a good way to learn, but it was also a way that there were a lot of things that weren't quite as reproducible as you might like, because the cells would change, the methods, you never have 100% of the things written down. And so Art and I worked on the ACTH receptor for a while, but we could never quite get it to work again. The cells had changed. One of the reagents you couldn't buy anymore. So I was very fortunate that at the same time, Pierre Fauché had been in Jesse's lab working on the insulin receptor. And it was just beginning to work with the insulin receptor. And so we figured that this was a good way to go. So I jumped in and started to work with Jesse and Pierre on the insulin receptor. David Neville was a very important collaborator in all of these studies. And it was a very robust system. It was a system that was quite reproducible, quite easily quantifiable. And because of that, we could really do the kinds of studies to not just characterize the receptor, but to do these pathophysiologic studies, look for changes in disease when receptors might have reduced binding or increased binding. It still was very much at an early stage, and everything that was going on was really like the first time, so I think that, Jesse tried to give some lessons to people. And one of the lessons I think I really learned from this is, and I learned a lot from Jesse in this regard, and I think Jeff and I spent many hours on this, is to try to look forward. There wasn't much you could look back at. There wasn't a lot of literature that you could say, this is what Dr. X did, we'll just do a variation of that, because we were right at the edge. And all of this work was really shaping a new field. And so we really had a chance to look forward to questions like how are things regulated? How are the receptors regulated in disease? Are there new receptor mediated diseases, just like there are hormone mediated diseases? So it was truly exciting. And it turned out to be both productive scientifically, but really, I think, a way that set the field in a very important direction for the future.
So I'd love to ask you a little bit more before we turn to Jeff and patients about your work in animal models.
Yeah. So animal models have always been important in metabolic disease, and they continue to be important, certainly in diabetes research. So we were able to use animal models for obesity and obesity linked to diabetes. But I would say this, that the ultimate animal is the human, right, in terms of human disease. So as Jesse said, we were able to work around there a little bit with human circulating blood cells, but so many of the insulin responsive tissues like liver or muscle or fat certainly are not easily accessible. So I think that the disease modeling in animals was very important, continues to be very important. One of the things that's really, I would say, happened over the last decade more that wasn't possible then, of course, is now we can also get humanized animals, and we can get human cells in culture through either iPS cells or iPS-derived cells that will mimic human. And that, I think, is a really powerful tool that, of course, didn't exist then. But by using animal models and then human cells, and now these even more advanced human model systems, I think we can really show that there is a lot of similarity between these disease models in animals and humans, but there are also important differences. And we have to keep those in mind and try to understand, what does that tell us about human disease? Because ultimately, that's really the disease we want to understand, right? Human type 2 diabetes, human obesity.
So Jeff, I want to now put you in the hot seat. So Jeff, I haven't really had the chance to interview you before, and so want to hear a little bit more about how your trajectory looked like in terms of what it was that led you to joining Jesse's lab at the NIH.
Oh, okay. Well, I can tell you that. So I went to medical school, and I was in the first class at Mount Sinai Medical School. It was a very small class. We got to know our faculty very well. There were only 36 students. And I ended up having two things that I decided I really enjoyed. I enjoyed a lot discussions of diabetes and insulin because we had, as professors, Solomon Burson, who was the chair of medicine, who was Jesse's mentor, and we had Panos Katsoyannis, who was the chair of biochemistry, who was leading the world in trying to synthesize the insulin molecule, and several other people. So I really liked that area. I used to hang out in some of the labs. And then at the same time, I got interested in a research project where I did some research in cellular immunology with my mentor at the time, Kurt Hirschhorn. And I published a paper in that area. And this was at the time, as Jesse said, the Vietnam War was raging by the time I was in medical school. And I thought of applying for the Public Health Service to go to NIH. And the way it worked, they had these matches where you could pick laboratories, and then they would interview you or not. And then at the end, you get a note saying you're either in or not and what lab you're in. So I had about 12 immunology labs that I applied to, many very famous immunology labs, and maybe 2 endocrine labs. And the endocrine lab was the diabetes branch that Jesse was heading. And I'll never forget, I went really just a few months before he died to meet with Professor Burson, who I'd gotten to know a little bit. And I said, can you give me some advice? I have to choose where to go or if they take me. And I said, I'm interested in immunology. I'm also interested in insulin. And he stopped me right there and he said, okay, there's only one lab that you should go to. You've got to work with Jesse Roth. He's going to transform the biology of how molecules influence cells. And whether you're interested ultimately in immunology or endocrinology, you work with Jesse. I'm going to call him up after you leave the office and everything will be fine. So it turns out that's where I ended up, in the diabetes branch with Jesse Roth. And that's exactly how it happened.
Were you able to, like, did you know that you would start to get into receptor-based biology? What was your project when you first joined the lab?
So this is also a strange story, but it is the truth, okay? I got there with a little bit of research background, very little, on another area of research. I had never done research on insulin in any way. And when I got there, it was like a awe-inspiring environment. Jesse was the tower of power. And there were like 25 or 30 people, each working on different projects. Ron was kind of the rising star, a guy coming out of his training and was leading a number of important projects. And what ended up happening was Jesse gave me freedom. And he said, okay, think of a project. You want to come back and run it by me? I'm happy to talk with you about it. And what ended up happening between July and November, December of 1974, was I tried and failed at about 5 projects. I had ideas. I said, how about this? I would get some reagents. I would try. And literally, I was more unsuccessful than you could have imagined possible. And I was beginning to think about leaving to go back to clinical work at where I came from. How I ended up working on a project that got me into research, I could tell you, but it's a short thing. So
I would love to hear this. Like, where did your aha moment come from?
Yeah, it was really simple and explicit. So we used to have Friday afternoon clinical rounds, and the NIH was the clinical center. So there was a corridor where we had patients who were referred for all kinds of disorders, including problems of insulin and growth hormone and things that Jesse and Phil Gordon were interested in. And then another parallel set of labs. And I had a bench in a lab, but Friday we would go to clinical conferences. And I went to one of the clinical conferences, and what was presented was a patient with severe insulin resistance. And they were there because they had severe insulin resistance. And the fellow who was presenting the clinical side of it presented the case and said that they seemed to have very low receptors measured on their blood cells. And I was listening to it. People were asking questions. But that particular patient whose case I heard had what I interpreted to be autoimmune features. And nobody even mentioned the autoimmune features that I can recall, or if they did, they didn't connect it to the disease. So I was about to raise my hand and say, what about an immune basis for the insulin resistance attacking the receptors? But I didn't, because I said, well, if I say that, someone will just do it. Let me just find out if anyone is doing that, So I kind of bided my time and I waited. I asked a few people questions over the next week or two, and there was no evidence that anyone was considering that line of attack. And at a certain point, I went to Jesse and I said, Jesse, my work hasn't been going anywhere. I have this idea, and I'd like to test the idea that there are some autoantibodies attacking the receptor. If there are, that would be really cool, wouldn't it? And he told me, yeah, that's a good idea. Why don't you work on it? So that's how I did it. And I was given the opportunity to pull together some sera and some cells and do the experiments. And it showed that took months to prove that they were autoantibodies, but that's what eventually happened.
So let me ask there, because one thing that the three of you have in common is you're medical doctors. And so, Jesse, how much were you and Ron and Jeff involved in treating these patients that had severe insulin resistance? And
we actually went out to look for them when let me go back 2 steps before the experiments that Jeffrey so beautifully described. We kind of said, let's find some patients who are very resistant to insulin. And we had good relationships with the diabetes community. So within a relatively short time, we got 6 patients who had extreme insulin resistance. It wasn't the insulin resistance that would come from having high doses of insulin. This was really astronomical. Let me jump then to Jeffrey's presentation. He said, well, maybe some of them have antibodies against something. Oh, that's a good idea. Well, anyway, when we actually got down to it, 3 of the patients had antibodies to the insulin receptor, blocking the receptor, causing resistance. Another 3 had inborn errors of the insulin receptor, blocking its action. And so, in fact, what was a miracle is before these experiments that Jeffrey described for you, we did not distinguish between these 2 groups of patients. And once you saw the 2 groups of patients, idiots, how did we miss it? One of them was clearly autoimmune, as Jeffrey described, and another group was clearly inborn and got it earlier. But it was this, the excitement at the NIH in those days is that the lab and the clinic were physically close and emotionally close. And the excitement of these findings is, I think, even now, and years later, still reminding us of how we kind of stumbled into these exciting.
So let me just make one other point, which maybe unites a number of the different lines here. So I have always said I was unbelievably fortunate after being unable to get an experiment to work to finally get myself in a position where I could have an experiment that worked, that was meaningful. The good fortune came from the fact that Jesse had established this field from nothing and became an advocate and a leader in the field, that he, with many other people, and Ron being a leader in that area, were characterizing these molecules as biochemical entities that you could study kinetically for how they interacted with the hormone and relate that to, ultimately what would be the biology of the hormone. And then they began to collect, various diseases, including these rare diseases that were sent to NIH because people knew that this was the home of the insulin action at that point in time.
Yeah.
And the techniques for measuring the receptors on the patient's cells or on other cells had been established. And I had the good fortune of just having this realization that a subset of them might well have antibodies. And then it was straightforward for me to show that there was something that looked like an antibody. And then it took a year to characterize what they actually were. And then with all of my colleagues, including Ron, it became possible to then use these antibodies as tools. Because I'll make one last point and then be quiet, is that, today, if you talk to scientists training in molecular science and what they now call signal transduction, everybody, just orders antibodies to things. Antibodies are what you have to play with, right? There were no usable anti-receptor antibodies at that point in time. And some of these patients had extraordinarily high titers. That enabled an almost infinite number of experiments to characterize the receptors in ways that Ron and many others did, none better than Ron. So I want to say that that's how these things came together.
And maybe I can just elaborate on this point, because I think this is really hard for people maybe to imagine. But these patient-derived antibodies really became the tool to understand the receptor before. I mean, the receptor hadn't been purified, it hadn't been cloned, nobody knew anything about it. But using the patient-derived antibodies, we were able to do biosynthetic labeling, determine basically the receptor structure, the fact that there was a pro-receptor that was cleaved to give 2 subunits. We were able to look at that process and really figure out the basic stoichiometry of these subunits. We were also able to use those same receptor antibodies to do immunoprecipitation to show that the receptor was a tyrosine kinase that underwent autophosphorylation. So ultimately, when the receptor was cloned, which was about, I guess, 1985, that it was published, the cloning of the receptor, we knew that the receptor was from a single chain precursor. We knew it was a tetramer with alpha and beta subunits. We knew it was a tyrosine kinase. And the cloning simply confirmed all of those things. It didn't, it obviously gave the sequence and it gave much more detail, but taking advantage of this was really an important thing. And maybe I'll give one other anecdote because I think it's, again, another useful way. I remember very well after Jeff made these really important experiments showing that these patients had autoantibodies to the receptor. At that time, there were 2 other presumed antibody to receptor diseases. One was myasthenia gravis with antibodies to the acetylcholine receptor, and the other was hyperthyroidism, Graves' disease with antibodies to the TSH receptor. The antibodies weren't really known to be to the receptor because, again, those receptors hadn't been cloned, but it was known that the TSH stimulating the antibodies in Hashimoto's or Graves' disease, would stimulate the thyroid, and the antibodies, there was an antibody in myasthenia gravis that would block signal transduction across the neuromuscular junction. And so this became like the third disease, although I think in some ways the leading disease, because it was able to be kind of a tool to really dissect the whole system. The third disease with anti-receptor antibodies. And at that time, in a very typical fashion for this kind of creative group that was assembled under Jesse, Jesse's first reaction was, well, let's get together, always on a Saturday morning. It was always, we get the call on Friday night. My wife would say, oh, no, we're not. You're going to be gone tomorrow morning. I know it for sure. And we'd say, okay, come on in. We're going to talk about this area. And the idea was to brainstorm about, well, what other diseases could be anti-receptor antibody diseases, and to think as broadly as we could. And we put a lot of interesting possibilities there, some of which have shown a little promise over the years, but none of which I guess have grabbed hold. But, we were encouraged to think big. And we thought, well, maybe essential hypertension could be an anti-receptor antibody disease. Maybe diseases like major mental illness, bipolar disorder, or schizophrenia could be anti-receptor antibody diseases. And several other anti-receptor antibodies have been observed to the gastrin receptor, to the gonadotropin receptor, and subsequently. But I think this is still an area that has potential in some other disease mechanisms. I do think, sort of going back to your point, that being physician scientists, this is one area where you have a slight advantage. I mean, the PhD scientists are certainly, and were certainly at that time, and still certainly more technically trained, more knowledgeable about various experimental procedures and basic mechanisms. And what we brought was understanding of pathophysiology, or the questions that pathophysiology left open, where you had, and so to think about bringing these two together was really the strength of this group in many ways. There were many other MD investigators and some PhD investigators, but we always were looking for the connection to either physiology or pathophysiology, right, and when possible, in people.
And actually, I'll add one other anecdote, which is, to me, an interesting story, which was, of course, the patients were discovered as being insulin resistant. And as was described initially, 3 patients of the initial 6 were shown to have these circulating antibodies that would block insulin binding, that were then characterized in various ways as antibodies. But the expectation was that if we would add these antibodies from the patients to cells that were responsive, they would just block insulin action. So just to show you how science doesn't always work the way you expect, we collaborated with Ron, who was at that point already making isolated rat fat cells for various studies that he was doing. And we added the patient's serum to the fat cells. And what was found was that it actually massively mimicked insulin action. It was a insulin agonist. And it took 6 months to a year of research by Ron and others to figure out that when you just added these, even from a very insulin resistant patient, you added it to a fat cell, you stimulated glucose uptake and oxidation. And then it was eventually shown, and I'll remember these studies like it was yesterday, you take the antibody molecule and you split it into the 2 FAB fractions. And if you add the FAB monovalent fraction, it completely blocks insulin action, but the bivalent stimulates. And that was one of the earliest indications that cross-linking sort of allosteric effects of receptors were important for insulin action. And then, in the ultimate irony, in a way, I was on the ward at a certain point when the original patient, I won't mention her name for HIPAA reasons, although she's passed away, 50 years ago, but she was readmitted to the hospital with profound hypoglycemia, low blood sugar. And they gave me her serum. And I immediately went to the lab and set up an assay to see maybe her antibodies went away. No, they were the same titer that they were before. So it left this conundrum. Why did someone who was unresponsive to insulin and high blood sugars evolve into a patient with hypoglycemia? That's a longer story, but it turns out there is a version of this disorder where people have low blood sugar, probably due to the agonistic action of the antibodies. So I'll stop there and see if Ron or Jesse have different ideas about those events.
So I wanted to ask you a question, all 3 of you. When you started this out, as Jesse had noted, everybody thought that there wasn't a receptor for insulin. So what was the reaction to your findings? Were you lauded? Were you laughed at? Was it a heretical thought?
Well, for sure, there was pushback on the idea of receptors being regulated. I think that People pharmacologically had talked about receptors. People had thought that there must be receptors, although there was this notion that insulin might enter the cell and directly interact through its disulfide bonds. That was, I think, all one theory, but there were certainly plenty of other suggestions that there were surface receptors for other molecules that could be present. I think that part was not so hard to get accepted, but what was hard was for sure the idea of regulation in disease. I think that everybody assumed that in endocrinology, hormones go up, hormones go down, signaling goes up, signaling goes down. And I think that what became very clear, and Jesse was a big proponent of this, but all of the work that we were doing and others in the lab were doing at the time really supported this, which is that the receptor is an equal partner. And the receptor too can go up, or the receptor can go down, or it can change its affinity. And one of the really striking things, and I remember this was actually a big debate even in the diabetes branch, was Jesse said, well, maybe it's insulin itself that's regulating the receptor, right? Insulin itself that's regulating the receptor. And Jim Gavin, who was at that time a fellow, was really the person charged with studying this concept that is insulin regulating the receptor. And we would have these data groups when he would show his data about insulin regulating the receptor, and we'd all say, are you sure? Maybe it's just, insulin is blocking the receptor binding. We were giving him all, and even to this day, he said, how hard it was to defend. But then once, of course, you got out of the group, you were ready for the outside. You
right.
had your toughest criticisms. But I think that part of the story took longer, but pretty soon people realized that this wasn't unique to insulin, and that people were studying regulation of gonadotropin receptors. Kevin Catt was a few floors away doing that, and people were doing glucagon receptors, glucagon signaling. So all of this kind of caught on. And as you already sort of know, the NIH was just kind of a remarkable hotbed of science, and signaling and peptide hormone signaling in particular was part of the hottest part of that hotbed, I think, during the period from sort of 1969 or 68 through about 1980.
So let me just add my bit to that. So when I got there in 74 and surveyed the scene, and everyone gave me their manuscripts, and I'm reading all the manuscripts and sitting in on the data meetings, to me it was established that what was being measured in the lab, that they learned how to measure, was a critical component required for insulin to act. So I drank the Kool-Aid. I accepted it from the moment that I began to pay serious attention to it. But what were the questions that were dominant, that weren't answered at the time that I got there? Of course, there were a million of them, but two that were dominant were, does it really play a role in disease? As opposed to, well, in this condition, it's a little lower. Yeah, but how do you know that is why you have a problem with insulin action? And then the second thing was, exactly how does insulin binding to the receptor cause anything to happen? And, for the whole time that I was there for 4 years, it wasn't yet, possible to say how insulin caused enzymes and transport to change. That required things that happened a few years after that with the kinase, autophosphorylation kinase pathways. And as far as, how do you know that it actually is critical for disease? It really was these extreme insulin resistant patients. And the ones that we now know have some kind of inherited mutation. In some cases, we know what the mutations that led to receptor malfunction were, but we didn't know at the time. We just know that there was low binding, right? The antibodies were really the final, proof that something that blocked, that came as a disease process, was cause you to have tremendously severe diabetes. So once that happened, I don't think there was any pushback anymore that this was a critical component. But what was required was to figure out how the normal receptor worked. And that's what Ron spent his career working on.
Let me go back a little bit if I can. Ushma, this is Jesse again. It was funny because I had visited a bunch of labs and came back with the idea in my head that if we had high insulin levels and low insulin responses, that maybe it could be that the insulin itself was driving the insulin resistance. And when and Jim Gavin, we used to have a data club you had to present your experiments for the week that next week. You couldn't wait 6 months like some places did it. You did an experiment, you presented to the group. And so Jim Gavin used to do the experiments that he and I planned, and then he'd come and present the data, and the group would howl, how do you know, but what about? And they would jump up and down. In fact, one of the guys from abroad said when he came to the lab and saw everybody yelling at Jesse, he thought we're on the brink of a major revolution. No, this is a regular meeting. So week by week, we would get more converts to the idea. But it was funny where one particular meeting, Jim said to me, look, Jesse, I'm not going to be beat up this week. You tell them the experiment and you get beat up. So in fact, it was fun that in fact, during that whole period of time, there was free play of good ideas. And it was that kind of open expression of ideas, I think, that helped us move as fast as we did, because everybody felt comfortable telling what they really thought.
So I'm going to actually ask a question of Jesse and Ron that reflects my own opinion on one thing, which is, so I came there in 74. That's 46 years, 47 years from today, right? And we learned a lot during that period. But this is my statement, okay, is that in 2021, we still don't know in molecular detail the basis for the insulin resistance that type 2 diabetics have. We know a lot of facts about it, but we can't answer the question. This is why people who are born and at a certain age, they develop insulin resistance and type 2 diabetes. And if you had asked me 40 something years ago, or even 20 years ago, that we would have the tools that we have, the genetics that we have, the biochemical reagents that we have, the longitudinal studies that we have, if you had said to me, what's the possibility that we won't have figured that out by 2021, I would say it's close to zero. But I don't think we have figured it out. We have a lot of facts, a lot of interesting insights, but we don't have the answer to the question.
Jeffrey, I think that you're right. And it's interesting, too, because every few months, somebody comes out with a paper claiming to have found it. And I read the paper quickly and shake my head and say, uh-oh, didn't do it. So I'm with you. I read the articles with excitement, but with continuing skepticism that they have, in fact, found the answer.
Well, I'm more in the middle of this, I guess, than Jeff and Jesse at the moment. I actually, first of all, I do agree that the most important disease for which insulin resistance is a component, or a group of diseases, are type 2 diabetes and metabolic syndrome. And this is a huge public health crisis of diseases that cause long-term complications and so forth. And at the core of them is certainly insulin resistance, either as the primary driver or at least a primary driver among several drivers. I think that there's no doubt about that. And I think it's fair to say that you're right, Jeff, in saying that we don't really know the absolute cause. I do think we've made a lot of progress trying to dissect the various mechanisms of disease. And there's really a lot of course, beautiful work that's been done by many laboratories looking at external drivers of insulin resistance, inflammation, fatty acids, circulating branch chain amino acids, and various factors which are dysregulated and can drive the cell to become insulin resistant. But in addition, and you know that our recent work has also focused on the fact that using iPS cells from humans with type 2 diabetes made into muscle in vitro, you can show that there's also a cell autonomous defect. There's something in that cell itself that's already resistant even before you add all these external factors. And this is, to me, the real challenge, whether it's extrinsic or intrinsic, is what is the driver. What is the driver? I would say here is a real challenge for not just diabetes, but for all the most common polygenic diseases. Genetics has been instructive and identified many loci that are linked statistically to disease, but has really, in only a very small fraction, been able to identify how those genetic loci actually create a pathophysiologic mechanism. And in type 2 diabetes, we really have to try to understand. And I think that we're at the brink of this, not me personally, but the field of science is at the brink of this, because we're beginning to think beyond GWAS studies. We're beginning to think beyond one gene or even groups of genes at a time into the whole 3D organization of nuclear chromatin and DNA and regulatory things that are changing gene expression. So I guess I agree we haven't come as far as we should. It's been 45 or 50 years. We got some smart people that should have figured this out, you think. But I think we are getting there, and I think that the same challenge, we still don't understand what causes hypertension. We still don't understand what causes most people to have atherosclerosis. Most people don't have LDL receptor defects. Most people don't have gene mutations like Rick Lifton has shown for some of the extremes of hypertension. These are syndromes, and the common varieties are the challenge. They are the challenge both as a public health and individual health problem, but also as a science problem. And there's a great challenge for somebody, to figure it out.
Yeah, well, I wasn't in any way trying to diminish that there has been massive progress, but I think even you would agree, Ron, that you would have thought 20, 30 years ago, we would have it closer to being nailed than we do today. Yes. And it will be figured out. So we will figure it out, but we don't have it yet.
I agree.
So I'm mindful of the time, and I have one last question that I want to ask all of you. And I know that we could stay on here for another hour probably, but what I want is a personal reflection from any of you. So could you tell me a story about what it was like being in the lab with these colleagues that are on this Zoom right now? Jesse, do you have a recollection of what, Jeff from 46 years ago and Ron from 46, 47 years ago were like in your lab? Did they come in with like a lot of chutzpah? Did they come in and do whatever it was you said?
I'll give you a quick anecdotal example and I'll close. Is that some outside company was doing a movie about NIH, and they came to my ward where we were doing rounds, and I was leading the rounds, and there were several others. And when he was interviewing me, during the rounds, no matter what I suggested, one of my colleagues jumped on me and told me out in the group why that was not a good idea. And so idea after idea that I proposed was shot down by one of my young people. So when he came to the interview, he says, Jesse, aren't you afraid of all the yes men that you have around you? I said, oh, God bless you. Find me one yes man.
Yeah.
So, but what I would like to say is that as a lifetime experience, I look back on all the wonderful people that I had working with me, and the great respect we had for each other and the dynamism of it. Wow, I couldn't trade it in for the people, their families, their futures, their own offspring. It's just been a hell of a good run. And I can't think of anything that I would replace it for. And those of you who've been lucky to have that kind of experience, wow, you've really been very, very lucky like I have.
So Ron, why don't you go next? Do you remember some particular story that will give us all a flavor of what that time was like in Jesse's lab?
There are so many stories that were truly remarkable, both scientific and personal stories, because it was like a big extended family group. Not everybody in a family gets along. Not everybody in the family is going to hug and kiss every time they get together. But there was a lot of mutual respect and a lot of love and a lot of pushback in many different ways. And I guess what, I could certainly say that, it's created lifelong, both collegial relationships, but friendships that are like family. And I think that, since Jeff and I live only a few blocks apart in Newton, we are as close as any brothers could be, I think.
Thank you, Rob.
Well, I'll just add then that, I'll endorse everything that my colleagues and friends just said. Jesse set the tone for a scientific community that was far more than a scientific community. It was a community of friendship and respect. And as Ron said, over the years from when I first met him in '74, we ended up being neighbors and colleagues. I was the dean of the school and he was a faculty member. And I will tell one brief anecdote because I don't think I've ever told this story, but it's a good story. So I came up in '78 to be at the Beth Israel, and Ron was still at NIH. And then I was the head of the diabetes unit at the Beth Israel. And then I knew there was a search for a research director at the Joslin. And by the time that was coming to fruition, Gene Braunwald, who was the chair of medicine by then at both the Brigham and the Beth Israel, was leading the search. And for the first time ever, he called me into his office to talk with him. I didn't know what it was about. And he said, I have a question for you. We're down to 2 finalists for the head of research at the Joslin. One is Ron Kahn, who you know very well. The other is someone else whose name I won't mention. And, I would like to know who you would rather have, because many people have conflicts with people they used to work with. It might be considered that you would be in conflict with him. So what do you think about Ron? I said, Ron's your man. He's the better scientist and he's my friend and you should bring him. And I don't know if that's what did it, but it certainly played out that way. And I've never regretted it for 1 second.
Nor have I.
It's a beautiful note to end on. Thank you so much, Jeff, Ron, and Jesse, for joining me for this hour. It's going to be really hard to cut anything out of this interview.
Ushma, thank you so much for taking the trouble
Yes.
to bring 3 friends together to have a marvelous afternoon,
Right.
recalling the olden days.
All right.